Hey guys, so this is the all trait practice. I'm just going to go through it. You can fast forward to see the questions that you have questions about, but here we go. So the first one's nice and easy. It's just a sex-linked trait. All right, so basically you got to figure this out. So we have a mother with an X-linked dominant. Now we haven't really talked that much about what difference it is on a dominant, but we're going to put the mom down the side. So here she is. She has the trait. So I'm just going to put a little hash mark on the X that has the trait. Because it's dominant, she only needs one X to actually show the genetic disorder. Now, she has a child with a male who does not exhibit the order. So he will have an X and a Y with no trait hash mark on the X chromosome that they do have. So let's take a look. We'll do this cross. Okay, we'll do this. This one's going to be a normal female. We have an affected male. And we have a normal male. So it's basically 50% of the females will have the trait, and 50% of the males will also have the trait. Now, interestingly enough, this asks a question, does it matter what the gender is? And the truth is it doesn't. If I were to do a normal Punnett square with this, okay, let's take a look. If I just do, I don't know, I'll use the letter G. Okay, so here's the dad, here's the mom. Okay, and the mom, oh, hang on. If this is the dad, he would actually be recessive, right? Because he doesn't have the trait. And we'll say the mom is heterozygous, right? So in this case, we would get 50% with the trait and 50% without the trait. And as you can see, that's exactly what we got there. So the gender doesn't matter, but that is how that question would be answered. Okay, next question is also a pretty easy question. In this case, they are carriers of a recessive genetic disorder. They want to know what percent that their chances that their child will have the disorder. So I'm just going to go, so in order to be carriers, they have to be heterozygous. So I'll just use H's here. In order to have the disorder, they would have to be little h, because this is a recessive disorder. So both parents are going to be heterozygous. And really the only place you're going to see an affected child will be 1/4. So they have a 25% chance of having a child with a disorder. They have a 50% chance of carrier child, which would be heterozygous, which you can see in these two squares, and they'd have a 25% chance of what we call normal, which is a horrible word, but that's what it would be. So that's your answer there. So this one's a little trickier. It seems kind of easy, but there are some options here that could happen. All right, so the first thing you want to do is cross a red flowered plant with a yellow flowered plant. So let's use the letter R, and we're going to let's assume that red is dominant. Okay, so then if that's the case, these would be red, and little r would be yellow. Okay, so if I cross homozygous dominant with homozygous recessive, and I'm assuming that red is dominant, then I should get 100% red on the F1 generation if red is dominant. Okay, but that's assuming that the red colored rose is homozygous dominant. Because what happens if it is not? If that's the case, you're going to get 50-50. So if you get a 50-50, so I cross red with yellow, I still am going to know that red is dominant in this case, because in this case I would get 50% red and 50% yellow. Okay, if yellow were dominant in this cross, you would actually still see 50%. So that's a tricky one. You have to know what the parent is. You basically have to do a test cross. And if you remember what a test cross is, you're basically going to take this F1 generation and cross it with a plant that is definitively homozygous dominant. or recessive, I'm sorry, recessive. Definitively recessive. Okay, so for example, let's say that I did say that red was the dominant color. That's what I'm thinking. I'm going to cross this F1 generation with, or let's say I cross this one. So I'm going to cross that, and then with, sorry, I can't talk, with a homozygous recessive yellow, and I should get 50-50. If that's the case, then I know that it's a heterozygous parent. It's still red. I know that red is dominant. All right, so that's how you're going to determine it. The test cross is going to be key. When you do a test cross, you have to cross F1 generation with a known homozygous recessive. It's also really important to note that we are assuming that when we cross these, watch your self-pollination, right? Like if I'm crossing plants, I have to make sure there's no other pollen going on. So that's also an important thing. That's all I want to say. So if it does produce some yellow, then it has to be heterozygous for the dominant trait. I hope that helped on that question. All right, so this question is looking at multiple alleles. And so let's try this out. It's going to be a little tricky, but just stick with me. I think it'll make sense. If a rabbit is brown, that is dominant to all other alleles. So all you need is one capital B in order to be brown. Black coat is dominant to gray. So in this case, if a bunny is black, I'm going to call it little b with a G, because black coloring is dominant to the G, but brown is dominant to black. I hope that makes sense. And then in order to be gray, which is the least dominant, you would basically need two G's. All right. So when we look at what we're crossing, okay, we are going to cross a homozygous brown rabbit. So we have a homozygous brown rabbit with a heterozygous black rabbit. So we have the B and the G. So let's take a look at what that looks like. So I've got big B, big B, big B, little g. Okay, so I get brown rabbit. Oh, look, a brown rabbit and a brown rabbit. So basically we get 50% homozygous dominant. We get 50% heterozygous, but phenotypically we get 100% brown rabbits, because remember, anything with a capital G, or I'm sorry, capital B, that is dominant to all other traits. That was a pretty easy one, actually. Okay, next is a blood typing question. This is also a question about multiple alleles. Remember in class that we did talk about this a little bit. I'll do this one in red, to have the theme of blood. Anytime you have a little I, that is basically saying you do not have any antigens, any genes for antigens. So it basically is saying you have no allele for a protein. If you have a capital I, that is saying that's like a placeholder, and then you're going to put the letter above it that indicates what allele or what antigen you have the gene for. Okay, so this is asking, suppose parents with type A blood had a child with type O blood. Is this possible? Well, type A, if you look, can have two different possibilities. All right, so you could do this by doing two chi-squares. That's one, or I'm sorry, Punnett squares. That's one way you could do this. But if you know enough about gametes and how sex cells are produced, you know that this genotype can only give this. All right, and this genotype can give two. It can give this, or it can give this to the sex cells. If I have both parents with this, or even one parent with this phenotype or genotype, they can't even give the allele or the gene for O-type blood. So both parents would have to be heterozygous for type A. Okay, so if you do that cross, you end up with a 25% chance of an O-type blood. So it is absolutely possible. But both parents, even though they're both type A, they would have to be heterozygous type A. All right, the next one is looking at basically kind of a dihybrid, but not really. So let's take a look. I've put all the phenotypes and genotypes here at the top so you can take a look at those just to kind of make it easy. Whenever you have multiple outcomes like this, always write down what the genotypes and phenotypes are before you start. It's just going to make your life so much easier. All right, so tasting is dominant and so is brown eyes. So let's take a look at what we have in the question. All right, so a brown-eyed male who is a taster marries a green-eyed female non-taster. So the other key piece here is that if this is the dad, the brown-eyed male taster, they say that the father's parents, one of them was a green-eyed non-taster. A green-eyed non-taster is basically this. So the only gametes that they can produce are going to be recessive. So therefore, we know that the male in this cross has to be heterozygous for both traits, because one of his parents could only give the recessive alleles. So we know that. And then we know that that male is mating with a homozygous recessive for both traits. That's the green-eyed non-taster. So let's figure out what cross we're looking at here. You have to foil this one, right? Remember foiling? So the possible alleles that you could get from this, or the gametes, are this. Sorry, that's a capital T there. Just doesn't look like one. Big B, little t, little b, big T, and then little and little. Okay, so you have the 4 different possible alleles on a dad, but the mom can only give this. So when we do this cross, here's the mom, and then I'll put the dad across the top, and I'll put his alleles here. Big B, big T, big B, little t, little b, big T, and little b, little t. So let's do this cross. We get heterozygous for both. We get heterozygous for eye color, but recessive for taste. Sorry, that was a little funky. We get recessive for eye color, so green eyes, with but is a taster, and then we get recessive for both traits. All right, so those are your outcomes. So we have brown taster, brown-eyed taster. We have a brown non-taster, brown-eyed non-taster. We have a green-eyed non-taster. Sorry, taster. And then we have, like the mom, green-eyed non-taster. So we get a 25% chance of each of those outcomes. Hope that makes sense.
Okay, guys, the last question is about the fruit flies. And there's a couple of different ways you can do this. I'm going to do it by doing 2 different Punnett squares, and then I'm going to combine the outcomes. So the first thing we have to figure out is what are the different genotypes. Okay, so I'm going to do eye color first. Let's see, there we go, eye color. And that one is sex linked. So I know that if it has a capital R, it's dominant, which is red eyes. If it's little r on the X, this is going to be recessive, and that would be black eyes. Okay, so when I do this cross, we're basically crossing a male with red eyes with a female with black eyes. So if we look at the red eyes, the male would have to be capital RY, and the female would have to have 2 recessive R's. All right, so I'm going to cross this. What do we get with this? We get, the dad will be up here. Let's It's weird to think of dad flies. Okay, and I'm going to have X big R, X little r, X big R, X little r. I get males with black eyes. So basically, if you look at this, you end up with 100% females in this cross. All the females will always be red eyes. And all of the males will always have black eyes, which is interesting because you're basically flipping what the parents are. It's kind of cool. So I hope that made sense. So that's our first one, okay? So I should say 100% for males on black eyes. Okay, so now we do wings, and wings is just a regular old Punnett square. So vestigial wings is recessive. So those would have to be, I'm going to use the letter N, little n, little n. Normal, what we call wild type wings would be big N, or big N, little n. So I'm going to assume, because it's AP, they want to make it more difficult. So I'm going to assume a heterozygous on the parent because we basically are going to cross a male with normal wings. So I'm going to use this for him with a female with vestigial wings. Vestigial. So when I do that cross, I get, it doesn't matter about the gender. I'm basically just going to cross big N with little n, little n. So I get 50% of both. So I get 50% normal wings with this cross, and I get 50% vestigial. Now, I want you to think about the fact that up here, I'm going to change color here. Up here, we have 100% of each gender having different colored eyes, but here we have 50%. When you cross, you have to automatically assume that 50% of your offspring are going to be females and 50% are going to be males. So therefore, I would cross this in half and say half are females and half are males. Same thing here. I would say half are females and half are males. So basically what you end up with is a 1:1:1:1 ratio. All right, because you know you're going to end up with red-eyed females with normal wings at 25%. You're going to end up with black-eyed females with vestigial wings. No, I'm sorry. That has to be red. There are no black-eyed females. Red eyes with vestigial wings will be 25%. Then we go to the males. All the males, if you remember, were black-eyed. So we have black-eyed normal would be 25%, and vestigial black-eyed male would be also 25%. So therefore, a 1:1:1 ratio. And that's it. That's the whole worksheet. I hope that helped. Thanks, guys.